Hi, everyone. This week in class, we're going to discuss more efficient computational methods for working with very large pathogen genomic data sets, especially methods that can make phylogenetic inference scale much better with the size of our data sets. And even though I'm going to emphasize that these more efficient methods can be applied to truly massive data sets, including data sets of millions of sequences, really a lot of these more efficient methods might be practically beneficial in cases where we have much smaller numbers of sequences too, because they can make analysis of pathogen genomes just much more efficient, regardless of the size of the data sets that we have. But as you're all probably aware, the size of different genomic data sets has grown massively in recent years for the most well-studied human viruses, for example, like influenza and SARS-CoV-2. We can now have millions of sequenced pathogen genomes. And although these like really well-studied human respiratory viruses are a bit of an outlier in terms of number of sample sizes, we're seeing the same phenomenon for virtually any type of pathogen. For example, now the FDA and USDA sequences tens or even hundreds of thousands of bacterial genomes for common enteric or foodborne pathogens, including things like Salmonella and Listeria and Campylobacter. So it's not just human viruses that are subject to these very large sample sizes. Virtually for any pathogen that we might be interested in studying using genomic methods, it's probably becoming extremely likely that we might have to deal with data sets of size of hundreds or if not thousands. Um, so we need genomic methods or methods for genomic epidemiology that scale well with the size of these growing data sets. Um, and this is in contrast to many of the methods that we've explored in this past semester, which have largely focused on Bayesian methods for doing genomic epidemiology, particular Bayesian MCMC methods as implemented in BEAST. And while I think there was a perfectly rational reason why we focus so much on these MCMC methods, namely that they allow for probabilistic inference of evolutionary and epidemiological parameters while accounting for uncertainty in both the parameter values as well as the underlying uh, pathogen phylogeny. Um, these methods don't necessarily scale super well with the overall size of genomic data sets. And this really isn't an issue with the efficiency, the inherent efficiency of Bayesian inference versus frequency inference. It's often a case with the fact that for many of the problems that we're interested, when we're doing Bayesian inference, we're forced to use MCMC to approximate or sample from our posterior distributions, which I'll argue for a couple different reasons is just inherently inefficient. So in the early weeks of the semester, we already discussed how we can think about MCMC as exploring the posterior distribution of trees or other parameters that we're interested in estimating using a random walk that we can think about as analogous to sort of a blind robot exploring the space of trees or parameter space. And this blind robot is, of course, blind because it doesn't actually know what this landscape of trees looks like. Instead, to explore and sample from different parts of parameter tree space, what we do is we allow the robot to take small steps in a random direction or larger steps. But there's the trade-off, right? The larger we allow the proposals to be, the higher probability those larger proposals are going to move this robot right off of the sort of peak of parameters or trees with high posterior probability. So most of those aggressive proposals are going to be rejected. But if we make our proposals too conservative and we always just propose very small changes in parameter values or trees, it's likely that we're going to be very slow to explore the space of all possible parameters or all possible trees. Um, Even in the case where there's only sort of one mode to this distribution or one peak. And of course, if there's multiple peaks, the problem gets even worse because it might take us a very large number of steps to sort of traverse the valley between two different peaks of the posterior distribution. So all this means is that while MCMC is a great approximation and allows us to sample from the posterior distribution to compute posteriors that we wouldn't otherwise be able to compute, it's not necessarily computationally very efficient. Um, in almost all of the MCMC algorithms that we've discussed this semester, we're using a Metropolis Hastings step to basically propose new parameter values or new trees. And many of those proposals will simply be rejected, meaning we're often evaluating the posterior probability, including the likelihood of our data, many times without actually updating our parameters. And also, if our proposals are conservative, which they often have to be, it also means we might have to make many proposals to move to a different part of parameter space. Or another way of thinking about this is for many iterations of the algorithm, we might just get stuck in a given part of parameter space, which means that we often have to sample hundreds, if not thousands, of MCMC iterations in order to get fairly independent samples from our posterior distribution. So what is the problem here in terms of applying Bayesian MCMC to genomic data sets? Is it the fact that we have many samples, or is it the fact that we have many sites in our sequence? So just think about that for a second. What do you think makes this a hard problem computationally? The number of samples that we have, or the number of sites for each sample? Right. Well, I'm going to argue that it's primarily an issue with the number of samples in our data sets that makes exploring tree space using MCMC sort of difficult. Because actually, the computational performance of a lot of our phylogenetic methods actually scales pretty well with the number of sites in our sequences. That's because if we think about the overall likelihood of our sequence data given tree, we often compute this on a per site basis by basically multiplying the likelihood of observing each column in our alignment together. And so we have to calculate the likelihood of the sequence data at each site. But this means that at worst, the overall computational cost of computing the likelihood of the sequence data is going to grow linearly with the number of sites that we have in our alignment. And often the cost doesn't even grow linearly because many of the columns in our alignment are not going to be unique. For example, we might have many, many, many sites in our alignment where um, all individuals sampled at a given site, individuals sampled have the exact same nucleotide sequence, right? So we have sites that have unique patterns um, that only occur once in an alignment, but we often also have many site patterns that reoccur multiple times across an alignment. And we don't actually need to compute the likelihood of these non-unique site patterns over and over again, as long as we compute it for one site with, say, like all A's or all T's or all C's, we can reuse that likelihood over and over again for all sites that share the same pattern of nucleotides across individuals. We can contrast this though with how the computational cost of exploring tree space increases with the number of samples. And I think I showed you the same slide early in the beginning of the semester, but basically the number of possible trees, the number of rooted trees that we can have of a data set with a certain number of samples or leaves basically the number of trees grows super exponentially with the sample size. So the number of possible trees that we need to explore is huge, basically like astronomical for any reasonably sized data set. And not only is tree space getting exponentially or super exponentially larger and larger the more we sample, but each time we sample a new individual, we also have another branch in our tree, which has an additional branch length parameter that we'll have to estimate as well. Um, so MCMC methods for sampling from the posterior distribution of trees generally aren't very efficient at sampling from this posterior distribution simply because tree space is so astronomically large. It would take us a very long time to even explore a very small part of that tree space. So in practical terms, what does this mean? It means for say an alignment length of say 10,000 sites, which is might be quite typical for say an RNA virus. We can use MCMC to sample from the posterior distribution of trees in say hours for data sets with 100 to a few hundred samples. But it's going to take us days to analyze larger data sets with hundreds of samples to say maybe just a little over 1,000. And then for data sets with thousands of samples, it might take us weeks to explore the posterior distribution and sample enough trees to efficiently approximate the posterior distribution of trees. So we can just see that there's sort of like inherently diminishing returns with using Bayesian MCMC to reconstruct bigger and bigger trees. It's going to take us exponentially longer and longer the larger our sample sizes are. So the first thing we need is faster tree reconstruction methods. And in fact, we've already seen some of these before this semester. Of course, we can always use maximum likelihood methods to reconstruct trees as well. And of course, maximum likelihood methods are very similar to Bayesian methods. They both consider the likelihood of sequence data under a given tree. But with most maximum likelihood methods, what we do is just greedily search through tree space to find the best tree that can best explain the likelihood of our sequence data or maximize the likelihood of the sequence data. And I just wanted to point out that most biologists working in phylogenetics and related fields still use these maximum likelihood methods because they are very fast and very efficient. Um, Bayesian methods like Mr. Bayes in the green and Beast in the orange um, are, taking up a bigger and bigger share of the market over time. Um, but still the vast majority of phylogenetic analyses that get published are done with maximum likelihood methods. And that's because there's nothing, wrong with maximum likelihood methods. Bayesian methods might be theoretically preferred because they allow us to quantify the uncertainty of not only trees, but all of our other parameters in a very coherent way. But if we're interested primarily in just reconstructing the best tree that we can, maximum likelihood methods can do that well and be much more computationally efficient than Bayesian methods. So we already talked about this a little bit very early on in the semester when we started talking about RAxML. I think we even used RAxML to reconstruct maximum likelihood trees in the very first tutorial of the semester. And the way that RAxML and indeed most maximum likelihood methods sort of search tree space to find the maximum likelihood tree is that they first use a very fast approximate algorithm to obtain a starting tree. Sometimes this is done by neighbor joining, just taking sequences that are most closely related in terms of genetic distances and merging them over and over again to cluster the sequences into a tree-like structure, or through maximum parsimony, which I'll explain in a bit. But then from that initial starting tree, what we often do is sort of a greedy search where we always try and see if we can find better and better trees that increase the likelihood of the sequence data given that tree. And so we propose new trees through these like tree arrangement moves, like nearest neighbor interchanges and subtree prunery graft moves. And those new trees are sort of accepted if that tree improves the likelihood of the sequence data. And at some point, we stop once we can no longer find any trees that continue to increase the likelihood of our sequence data. Um, and the worry, I guess, with this approach is that it's a greedy approach. It always just sort of climbs the hill until it can't find a better tree. But there's no guarantee that there's going to be a single peak in our distribution of trees, right? It might be multimodal with many local maxima of different islands of trees that all have very high likelihood. So the way that maximum likelihood methods get around this is it's often recommended to start our search from many different initial trees to sort of demonstrate to ourselves that regardless of what tree that we start with, we obtain sort of the same best maximum likelihood tree. Some recent work has sort of compared the performance of different maximum likelihood methods. We, for example, have already seen RaxML, but PhiML is another very popular and widely used maximum likelihood method. And then in recent years, like in the past, say 10 years, there's been newer methods that have grown in popularity very recently, including IQ-tree, which not only reconstructs trees, but can do some other nice things like dates or time calibrate trees for you. And then FastTree, which, as I'll discuss in a second, is truly fast and uses a bunch of heuristic approximations to try and speed up maximum likelihood inference of trees as much as possible. So in this analysis by Zhu and al., they looked at sort of the run times that it took to obtain maximum likelihood trees for data sets of different sizes. Um, and here the run times um, are the runtimes of these different algorithms are compared against the runtime of RaxML as sort of a reference data point. Um, and so you can see that basically on a log scale, runtimes always sort of increase linearly. So that means with sample sizes. So that means, of course, on a linear scale, these runtimes are growing exponentially with sample sizes as we might expect. And all of these different methods have very similar runtimes, except for FastTree. FastTree is very fast because it uses very approximate searches to find the best tree. And we can see here that oftentimes FastTree can be 10 to even 100 times faster than these alternative maximum likelihood methods. But we might wonder if this comes with some sort of cost in terms of accuracy and our ability to find the best trees. Uh, and indeed it does, because this analysis by Zhu and al. Also looked at um, the ability of these different maximum likelihood methods to find sort of the best tree. So this is done with a comparison of different empirical data sets. So we don't actually, in this case, know what the true tree is for any of these data sets, but we can sort of see if a given method can find the best possible tree by seeing if these different methods, how often these different methods find the tree with the highest overall likelihood score. The idea, of course, being that the tree with the highest likelihood is most likely to be the true tree. Um, and all these methods, besides fast tree, do sort of like equally well. But you can see fast tree here in the bottom rarely finds the tree with the highest likelihood, suggesting that it might be a little just too approximate, that its speed up comes at the cost of being able to find the best trees. Um, Meanwhile, the comparison that's being made here between the darker colors and the lighter colors is comparing the performance of the other three methods, RaxML, PhyML, and IQTree, um, when just one search is done versus doing 10 independent searches starting from different initial trees. And you can see that like starting from different initial trees really helps the algorithm. All three of these algorithms more often finds the best trees with the highest maximum likelihood. So it does seem like there is really a benefit to be gained from doing multiple independent searches, starting from independent initial trees with these maximum likelihood methods. So in our group, I think because of the sizes of data sets that we often encounter now, especially for viral pathogens, where we might have thousands, if not tens of thousands of samples, we often use these faster maximum likelihood methods. I think RaxML and IQTree are like our current favorites. Um, IQTree is especially nice because it can not only reconstruct trees for you, but it can generate maximum likelihood time calibrated trees, which is really nice if you want to be able to sort of like date the events in a given tree, like the time of a most recent common ancestor, like we do in Beast, without necessarily having to do Bayesian MCMC. Um, Fast tree can still be useful. I just somewhat skeptical that it's accurate enough to say used for publication quality tree reconstructions. It can still be nice if you just want to very quickly obtain a tree for a very large data set. But we've grown to be kind of skeptical that FastTree is actually capable of finding the best possible trees when compared against these other um, fast ML methods. So another issue that we often encounter when we deal with larger and larger data sets is that these data sets will themselves be growing over time, right? So like, for example, during any sort of outbreak or even a pandemic, new sequences will always be coming in. And we can ask ourselves, like, do we actually need to reconstruct the entire tree every time we obtain new samples, especially, for example, if we already have a tree with 10,000 tips and we sample 10 more tips, we can ask, do we really want to like redo our entire analysis? And the answer typically is no. And actually, there's maximum likelihood methods out there, including methods that are available from within IQTree and RackSmell that consider what's known as the sample placement problem, which is the idea of like, how do we place a new sample on an existing phylogenetic trait? Um, for truly massive data sets, the sample placement problem itself can kind of be computationally intensive. Say, for example, if we had like a tree, like a SARS-CoV tree with 38,000 tips, and we wanted to figure out where to add just one new sample onto this tree, to do that under a maximum likelihood framework, we would have to compute the likelihood of the tree, considering all possible attachment points of that new sequence onto the tree. So we'd have to basically do 38,000 new likelihood calculations, which can take quite a bit of time. In this case, for a data set of this size, it could take almost a half of an hour to place one single tip on the new tree. So this has inspired searches for faster ways of doing placement. And one idea that sort of emerged, especially during the SARS-CoV-2 pandemic, is this idea of using these ultra-fast placement algorithms, which work on what are known as mutation annotated trees to try and find where a new sequence would attach by trying to find the attachment position that would minimize the number of additional mutations required to explain the sequence data at that tip. So in a little more detail, how this works is that we start with a reconstructed tree. In addition to reconstructing the topology of this tree, for every branch in this tree, we keep track of which mutations would be required to explain the sequence data at the tips. So, for example, you can see that for branch 2 here, in order to most parsimoniously explain the tip data observed at sample S1 and S2, there needs to be one mutation at position 7 to explain that data. So now we can do this for adding a new sample. We can ask if we say add a new sample, which in this case is S5, how many mutations would be required to add this new sample to each position in the existing phylogeny? And so for adding this sample in some positions, it would require two additional mutations to explain the sequence data based on what mutations have already been mapped onto the tree, or even three mutations, but there's at least one position in which only one additional mutation would be required to explain the data observed at the tips. And so this is what we call the most parsimonious reconstruction, the tree reconstruction that requires the fewest additional sort of changes or transitions, or in this case, mutations. So yeah, in this case, we would place S5 along branch 3 here because that minimizes the number of additional mutations required to explain the sequence data at all of the tips. So this strategy of using maximum parsimony and mutation annotated trees tends to actually work really, really well and be really fast, as long as the amount of genetic diversity in our sample is relatively low overall, and most sequences are separated from one another just by a few mutations. Um, but it's well known theoretically that like maximum parsimony does run into problems, especially for data sets that have more diversity. Because if we're dealing with more divergent sequences, it can be the case that the most parsimonious reconstruction is not always the most likely reconstruction. That is to say, sometimes there might be alternative histories that require more mutations, but might actually be more likely under a given model of molecular evolution. Maximum parsimony is far from perfect. But in cases where there's limited genetic diversity and it's fairly easy to tell where mutations must have occurred on a given tree to minimize the overall number of changes required on that tree to explain the sequence data, these maximum parsimony methods can really work quite well and speed up the placement problem by quite a lot. All right, so we have access to faster maximum likelihood tree reconstruction methods, and we can even use maximum parsimony methods to place new samples on those maximum likelihood trees. But as we've seen throughout the semester, oftentimes we're not just interested in trees, but we would like to get time-calibrated trees where branch lengths are in units of real calendar time, so we can see how far in the past um, uh, events in the tree, like particular branching events or common ancestors, occurred in the past. So we'd like to get time scale or time calibrated trees, but in order to do this, we need to estimate the molecular clock rate. So for many years, it was kind of the Beast was sort of the only game in town for doing this. If you wanted to reconstruct time calibrated trees, there wasn't much software out there besides Beast that would allow you to reconstruct time calibrated trees, would sort of like force people to do these time calibrated phylogenetic analysis using Bayesian MCMC. But there are other ways of estimating molecular clock rates, and then using those molecular clock rates to date the times of individual events in trees. We've already talked about one of these methods, and that's root to tip regression. So remember that if there's a constant clock rate or mutation rate, which mutations are accumulating along all of the branches in a tree, we would expect mutations to accumulate at a constant rate. Um, and that means that if we regress the root to tip distance of like how far our samples are from the root against the time at which individual sequences were sampled, we expect to see this linear relationship, right? Where like the farther into the future or the farther towards the present individuals were sampled, the more mutations we would have expected to accumulate since the root. And when we talked about these root to tip methods, the thing that we noted is that in this case, the slope of this regression line um, actually gives us an estimate of how quickly mutations accumulate per time. So it gives us an estimate of our molecular clock rate, which we can then use to convert branch lengths and units of mutations into branch lengths and into units of time. And so in recent years, or at least the past 10 years, people have begun to use this idea to very quickly date or time calibrate large phylogenetic trees. There's one very popular method for doing this called least squares dating, which basically turns the tree dating problem into a least squares regression problem. Um, and the way that it does this is that LSD, or least squares, the least squares dating method, tries to both estimate the heights of all the nodes of the tree, that's the T's here, as well as the overall molecular clock rate, which we'll call omega. And so the way that it tries to find the best fitting node heights and clock rates is it treats it as a least squares regression problem, where we can consider the deviation from basically the observed branch lengths in units of the number of mutations that occur along each branch or the substitutions that occur along each branch compared to how many mutations that we would expect given our estimate of the clock rate, omega, and the time separating parents and children in the tree. So TI would be like the time of a child node, and TAI would be the time of its parent node. And so the difference between those two times would basically give us like the branch length separating two nodes in the tree. And then if we multiply that by the molecular clock rate, that would give us the expectation for how many mutations would occur along that branch, which then we can compare to the actual observed number of branches. And using the principle of least squares, what we want to do is we want to minimize the overall discordance or difference between the expected and observed number of branches. And the estimates of the node heights and the omegas that minimize this overall discordance, or actually the sum of the square differences between the observed and expected number of mutations, will give us our least squares estimate of the clock rate as well as all of the branch times. So this is a quite clever way of turning a hard problem into a very simple regression type problem, but this actually tends to work pretty well. People have compared the performance of different tree dating methods, including least squares dating, to more sophisticated tree dating approaches, including using beasts to sample from the posterior distribution of time-calibrated molecular trees. And what we can see is that these alternative methods that use regression-based methods like Tempest and least squares dating often return very similar estimates of the molecular clock rate compared to the more like principled approach that Beast uses. There, of course, might still be an advantage to using Beast in that Beast is able to properly quantify all of the uncertainty in the clock rate, as well as all of the other parameters in our model. But if we're just interested in getting a a quick and fast approximate estimate of the clock rate. It's generally the case that least square dating and Tempest does a very good job of this for us in much less time than it would take Beast to infer time calibrated trees. Um, so there's sort of a suite of different methods that have come out for doing fast dating of phylogenetic trees. I already mentioned Tempest. If you're a Python user, you might be particularly interested in time tree because this is a Python package that allows you to actually do a bunch of maximum likelihood based phylogenetic analysis right in Python. But one of the things that does quite nicely is it can estimate clock rates and optimize branch lengths in units of real time um, using methods that also simultaneously reconstruct ancestral sequences in a phylogenetic tree. And if you're interested in bacterial phylogenies, one approach that has been developed is backdating, which kind of is also based on a Bayesian approach, but it kind of breaks the Bayesian approach into two sequential steps. So rather than trying to jointly estimate both the tree as well as the clock rate, backdating works on fixed phylogenetic trees. And then given the fixed phylogenetic trees, it uses Bayesian methods to try and estimate what is the posterior distribution of clock rates. And it's nice because it also has some methods that we talked about when we talked about clonal frames that can also account for the possible of effects of recombination. So if you're interested in doing time-calibrated phylogenetic reconstructions for bacteria, I would definitely also check out backed dating. So we talked about faster tree reconstruction methods and faster dating methods for reconstructing time-calibrated trees. But what we're often interested in is doing downstream analyses on those reconstructed trees. And as we saw many times throughout the course of the semester, one of the things that we're most interested in doing is reconstructing ancestral states on phylogenetic trees um, in many different contexts. For example, if we're doing any sort of like phylogeographic analysis, we might want to reconstruct ancestral locations on our phylogenetic trees. But these ancestral states can represent other things as well, including the ancestral sequences at tips or the ancestral hosts, if we're talking about a pathogen that can move between multiple different pathogen types. And when we talked about phylogeography and doing ancestral reconstruction in phylogeography, we talked about doing it inside beasts, so therefore using Bayesian MCMC to sample ancestral states. Um, which is again is nice because it can give us sort of full measures of uncertainty, both about those ancestral states as well as quantifying the uncertainty in the phylogeny itself. But if we think that we already have a good phylogenetic tree, there's definitely much faster maximum parsimony and maximum likelihood methods that can make doing ancestral reconstructions on large phylogenetic trees much more efficient. So we can first consider like how we would do ancestral state reconstruction in a maximum likelihood framework. It works very similar to how we computed the likelihood of sequence data at the tips of a phylogenetic tree um, under a given Markovian model of molecular evolution, um, uh, also considering the case that we don't necessarily know what the ancestral sequences are at the internal nodes of the tree. So when we talked about doing this for Markovian models of molecular evolution, we noted that basically we could calculate these transition probabilities that describe the probability of going from any one nucleotide or any base to any other base, given any possible substitution path. And so we could use these transition probabilities to calculate what is the likelihood of going from any um, ancestral sequence to an observed sequence at the tips of the tree. For example, if we wanted to compute the likelihood of the sequence data at this second site here, where this individual has a C and this individual has a G, we could sort of postulate that the ancestral sequence here was a C, and we could compute the likelihood of the observed data at the tips by considering the transition probabilities of going from a C to C at one branch and a C to G at the other branch to compute the overall likelihood of the sequence data, at least for these two tips at this one site. And maximum likelihood ancestral reconstruction basically uses the exact same idea, but now instead of considering there to be like any particular base here at the ancestral sequence at a given site, we can consider all possible bases, so all possible four nucleotides at this one given site. And then we can see which ancestral sequence maximizes the likelihood of the observed states or sequences at the tips, again, by using the transition probabilities. And here, this argmax just means that we're going to take the nucleotide X at this internal node that best explains the likelihood of the sequence data at the tips. And so the idea, if we wanted to do ancestral sequence reconstruction, is that we would do this iteratively for each internal node in the alignment, uh, for each site in the alignment as well. So there's also been some really nice software packages that have come out in the past few years that now allow you to do maximum likelihood ancestral state reconstruction, both for sequences as well as any other possible ancestral state, like ancestral geographic locations. Under general Markovian models of evolution, like the GTR model, one that we've become quite fond of and used a lot is this PASTML program from Oliver Glasgow's group, which is nice because it's pretty fast and allows you to do maximum likelihood reconstructions of ancestral states on even large phylogenetic trees. And while it doesn't take into account necessarily all of the uncertainty in the underlying phylogenetic tree like you would do in a Bayesian phylogeographic analysis, it does compute what we can think of as sort of basically like the marginal probabilities of the each node being in each possible state. So it does allow you to quantify some of the uncertainty about ancestral state reconstruction. PaSTML is also cool because it can also it has this nice visualization feature where um, you can simplify sort of like the phylogeographic dispersal of a given pathogen. So in a case like this, where you have many samples from different locations, including maybe some samples that are very highly sampled, what PaSTML can do for you is it can reconstruct simpler graph that which show the general pattern of how these viruses have moved from location to location without necessarily having to look at the full phylogenies underlying these inferences. So we already talked about maximum parsimony tree reconstruction when we talked about the ultra-fast placement methods. But maximum parsimony can also be used to reconstruct ancestral states in a very, very efficient way. And so maximum parsimony methods are really what we think of as sort of like minimal evolution models. That is, they try and minimize the number of transitions or state changes required to explain the observed states at the tips of those trees, where those changes might be due to, say, mutations, or if we're talking about a phylogeographic model, migrations. And maximum parsimony actually works very well when the rates at which changes are occurring are quite low. In that case, you can actually show that maximum likelihood and maximum parsimony reconstructions are consistent. You only really run into problems with maximum parsimony, as I mentioned before, when many transitions at a given site or many transitions along a given branch are possible. In that case, the most parsimonious reconstruction might not actually be the most likely reconstruction And it's known that maximum parsimony reconstructions can suffer from some issues with doing reconstruction accurately when uh, many possible transitions are allowed to occur or can possibly occur uh, over a given phylogenetic trait. Um, but if that's not the case, and like there's few transitions in states over a given phylogenetic tree, maximum parsimony methods work very well and are very efficient. The general way that they work are they use clever dynamic programming methods to reconstruct ancestral states, which I'll just quickly go over because it's fairly easy to grasp how these methods work. Basically, most maximum parsimony methods for reconstructing ancestral states on trees use a simple two-step algorithm where in the first step of the algorithm, we basically compute the number of changes or the number of transitions that would be required to explain the data at the tips at each internal node of the tree. So here, I've colored the tree corresponding to whether each tip is in one of two possible states. So state 0 is, say, on the left here, and state 1 is on the right. And so in this case, this individual would be in state 1, this individual would be in state 2, and this individual would also be in state 1. And so in this first step of the algorithm, what we call the post order step, what we do is calculate the number of changes that would be required to explain all of the tip sequences or tip states below. And so in this case, we can see that regardless of whether this internal node is in state 1 or in state 2, we would require one change to explain the observed tips because the children are in different states. So there has to be at least one change along one of these branches. And we basically walk up the tree performing the same computation iteratively over and over again. So at each node, we explain where we compute based on each possible state how many changes would be required to explain all of the tips below. So in this case, that's sort of the root of the tree. We can see that uh, if this ancestral sequence was in state 1, only one possible mutation would be required, right? And that's quite easy to see based on the tip states because we can see that like since this guy and this guy are both in state 1, we would only have one mutation required to move this guy into state 2. Whereas if this ancestor was in state 2, um, two different mutations would be required to explain the observed tip states, right? We would probably need to have a mutation on this branch as well as a mutation on this branch. And so what we can do then if we want to find the most parsimonious reconstruction of ancestral states over the entire tree is we can walk back down the tree in each internal node, pick the state that minimizes the number of changes required below that node. So at this ancestral or root node, we would pick state 1, and then we would also pick state 1 here because this would give us sort of the path of states from the root to the tips that requires the fewest overall number of changes, which along this path would just be one mutation most likely occurring along this branch. So maximum parsimony methods are very simple, easy to understand, easy to implement, and very fast because they use this very efficient dynamic programming algorithm that I just described. And there's no other estimated parameters in the model. They're actually sort of a parameter-free model in that there's no rates or any other sort of like transition parameters or substitution model parameters that we have to estimate when we're using maximum parsimony. So that's why they've sort of become a very fashionable method, especially as we've grown, as sample sizes have grown in number, and now these maximum parsimony methods also underlie other clever approaches that we've seen like the ultra-fast placement method that we looked at for tree, the sample placement problem in phylogenetic reconstruction. Okay, so finally, just wanted to mention that you know, it can be sort of painful if you have this beautiful large sequence data sets to like give up any of that data. I mean, generally we don't want to throw out data that we've worked hard to obtain. But what I'm going to argue in the last part of this lecture is that oftentimes it makes sense to subsample from larger sequence data sets. And also by subsampling or resampling from these larger data sets, we can also learn a little bit about how our inferences might have changed if we would have sampled in different ways, right? And so there's basically two things that we can consider, right? We can consider how we might subsample from a different data set to downsample the size to a data set to a more manageable size. But we could also consider how we could subsample from a data set to eliminate sampling biases. So if we sampled 90 sequences from one location and only 10 sequences from another location, we could downsample to say have 10 sequences from both locations to have a more balanced data set. And then we can use these resampling strategies. We can say subsample a large number of times to see how our inferences might change if we would have sampled in different ways. And so as an illustrative example, we'll consider SARS-CoV-2 again, because this is a case where we have extremely large numbers of samples. In fact, now well over 20 million samples collected from all over the world. But there's huge sampling biases here too, right? So like we have huge number of sequences from North America and Europe, maybe certain places in Africa and Asia, but there's other locations where we basically have very poor sampling from, especially relative to human population sizes. So during the early years of the pandemic, I was sort of tangentially involved in a project that was trying to basically understand how the pandemic spread um, globally around the world, and then specifically how the pandemic spread in Africa. Um, and the interesting thing is that actually overall, Africa was relatively well sampled during the pandemic. I mean, there were countries like South Africa who were doing a great job of sampling a large number of individuals. And that turned out to be very beneficial because a number of new important variants like Delta and Omicron ultimately were discovered in South Africa. Um, but uh, this actually was kind of created an interesting political conflict between, what we can think of as sort of like the global north versus the global south. Because obviously the global north has much more resources to do this sort of like pathogen surveillance. But meanwhile, the global south, including countries like South Africa, were doing the best they could to sample a representative number of SARS-CoV-2 genomes and doing really important things like discovering these new variants. But in the end, they were almost being punished for this effort because as soon as new variants were being discovered in places like South Africa, uh, travel barriers would go into effect, limiting the movement of, South African citizens to Europe or to North America. So like one can think about this as like they were doing the right thing in terms of they were being good sort of like global partners and participating in global genomic surveillance, but they weren't getting any benefits from that. They were actually getting penalized for looking for variants. So as part of this analysis, looking at how SARS-CoV-2 spread both sort of around the world and Africa, we were really interested in looking at like how much actual movement there were of viruses say from uh, outside of Africa, the rest of the world into Africa, and how much movement there was from Africa back out to see whether these travel restrictions were actually justified or not. And so in this analysis led by Ed Wilkinson, um, what we were able to do using all of the sequence data available from Africa and the rest of the world is quantify using phylogeographic methods how many different sort of like imports there were from any location to any other location. So here, for example, in the pink, we see how many samples moved from different African countries to other African countries versus the blue, which shows us how often samples moved from Africa to the rest of the world. And then another way of like visualizing the same data is we can look at the number of times that viruses were introduced into African countries through time. And we can see that like at the very beginning of the pandemic in like the early months of 2020, basically like all introductions into African countries were from uh, the rest of the world, so outside of Africa, which makes sense because, of course, the pandemic didn't start in Africa. It started in Asia and then moved to Europe and then eventually North America. So it makes sense that the virus needed to be introduced into these African countries. And then slowly over time, what we see is that more and more viruses were introduced between countries due to movement of the virus within Africa. Um, but actually, what this suggested to us is that during this critical period of the pandemic, it seems like actually um, the opposite of what the justifications for the travel bans were occurring, that like most of the viral movement was from the rest of the world into Africa, and there was very little movement of the virus from back out of Africa to the rest of the world. But of course, we can ask like, well, how does this all depend on sampling, right? If we would have sampled differently, if we would have sampled maybe like more in Africa and less in the rest of the world, we would have might have come to completely different conclusions in our phylogeographic analysis of how these viruses were moving around the world. And this is where subsampling and resampling methods can really be helpful because we can basically use subsampling to look at these counterfactual scenarios of like, well, what we would have inferred if we would have inferred if we would have sampled differently. Um, so in this case, our basic strategy was to um, first build maximum likelihood trees um, from the full data set, and then subsample or resample individuals from each location at a chosen subsampling fraction, and then prune all of the other samples from the phylogenetic tree. And then from that subsampled phylogenetic tree, we could rerun our phylogeographic reconstruction of how the pathogen moved between different locations. And from that phylogeographic reconstruction, then we could ask like how many imports or exports there were from each location into any other location. And what we found, perhaps not surprisingly, is that as we increase the sampling fraction, either in Africa or for the rest of the world, the number of imports into Africa only grew and grew and grew, suggesting that like the more and more we would have sampled from Africa, if anything, we would have found more and more imports into Africa from the rest of the world, and not necessarily more exports from Africa into the rest of the world. Um, so we were obviously only sampling sort of the tip of the iceberg here, and our samples were only revealing a small fraction of like the true number of viral introduced lineages that had been introduced into Africa over time. I was talking about this with Ed Wilkinson one day, and I was like, "Well, it's just the tip of the iceberg." But he mentioned to me that South Africans have this like really nice saying where instead of saying tip of the iceberg, they say the ears of the hippo, which makes complete sense if you've ever seen hippos in the wild. You might see some ears popping out of the window, but you really have no idea how much hippo biomass is actually lurking beneath the water. So I really like this idea that like when we're sampling pathogen genomes from locations where we're sampling huge outbreaks or epidemics anywhere in the world, we're really generally only sampling the ears of the hippo. And so there's a lot of undiscovered genomic diversity there, which could change and potentially um, uh, update or give us a different interpretation of what's happening and how these viruses are moving from location to location. But in this particular case, it seems that by subsampling and resampling from our full data set, we can actually show that like if anything, if we would have sampled more, the number of introductions into Africa would have only grown. And so for the tutorial on Wednesday, which we've decided is going to be completely optional, you'll get a chance to play around with these sort of subsampling and rarefaction analyses using a set of viruses, SARS-CoV-2 viruses, but simply from in this case North Carolina to see how if we would have sampled more or less in North Carolina versus any other state how our inferences about phylogeography and how many imports of SARS-CoV-2 there had been into North Carolina would have changed depending on how we sampled. And I will say that again, this tutorial is entirely optional, but it may be of interest to you, especially if you're going to be working with larger data sets in your future, or specifically for the independent mini projects, because this tutorial shows you how you can use Python as well as some other quicker bioinformatic tools like RACSML and LSD to do large scale genomic epidemiological analyses much quicker. So yeah, I mean, I think it's clear that like the size of genomic data sets for pathogens are only going to grow or continue to grow into the future, and so uh, the computational efficiency of these analyses is going to be the major limiting factor in a large number of cases. But what I hope I showed you today is that even though in many cases we can think about doing sort of like full Bayesian phylogenetic or population genomic inference as sort of like the theoretical ideal thing to do, which properly takes into account all of our uncertainty in the tree as well as our other evolutionary parameters. There's perfectly valid and very efficient methods for doing some of the same types of analysis, reconstructing trees, dating those trees, and reconstructing ancestral traits that are much faster and often just as accurate as Bayesian methods. But the only really downside being they don't allow you to fully quantify uncertainty in both the phylogeny and the other estimated parameters. And regardless of how big your data set is, I mean, these methods don't only apply to massive data sets with tens of thousands of um, tips or millions of tips. Really, these faster, more efficient methods can be applied to data sets of any size. And this can be quite nice for doing early sort of exploratory data analysis, right? When you first get your own sequence data back, even if you have dozens or hundreds of samples, often you want to try very many different things quite rapidly. So you might want to say like build a maximum likelihood tree and then do a fast phylogeographic reconstruction, just so you can get a sense of what's going on in your data set. And these more efficient computational methods allow you to do that very quickly and very nicely. And then, of course, you can always move on to more sophisticated Bayesian or other analyses to get you the publication quality results and analyses that you need. So I hope, I guess, in the end that I showed you that even though I've been sort of like arguing sort of for these theoretically, very idealized methods that we've often used in Beast throughout the semester, pragmatically, when I or anybody else in my group are analyzing data sets of almost any size, we're really mixing and matching these different methods and approaches and often combining much more approximate but much faster maximum likelihood or maximum parsimony methods with Bayesian approaches to actually try and fully understand what's going on um, in our data. All right, well, that's it for today, and we'll be back on Wednesday with the optional tutorial, or you can use the time on Wednesday to work on your independent projects. Right. Have a good day. I'll see you all later.